mutation works, why there is a specific color for certain mutations, and what kind of mutations we can combine and what kind of mutations we should avoid combining. Okay, so these are the three main topics that I'll be talking on. So whoever has doubts maybe can write down somewhere, and towards the end of the session, we can clarify the doubts. And if someone has doubts in between also, you can just stop me. I'll try to clarify that. Okay.
Sure.
So we will start off with the feather structure. So before, as I told you, right, before understanding any mutation in African lovebirds or any other parrots or any other birds, it is always important for us to understand what is a feather structure. And before understanding the feather structure, now first we should know what are the kinds of feathers that are available in African lovebirds or any other birds. Okay, and the feather anatomy, the basic terminologies that we use for different parts of the feathers. Okay, so here is a graphical representation of a feather of a bird. So you can see, you might have noticed all these things, but scientifically you don't know what are these things called and how the color of these feathers are yellow, red, or blue. Okay, so today we will try to understand this and then go to the INO mutation understanding. So feather basically has a hollow shaft which is called as calamus, and this gets extended and merged towards the end, and this is called as a rachis. And we have some fur kind of structure on both the sides of the rachis, which is called as veins. And the feathers at the bottom are called as after feathers, and the feathers at towards the extreme bottom are called as the down feathers. So now when we zoom a little bit onto the feathers, right, we see extensions. So the extension of the rachis, basically this is the rachis. The extension of the rachis is called as barbs. Okay, and the extension of the barbs, you can see there are two extensions for the each barbs that is coming out from the rachis. So these are called as barblus. Okay, and further when you again look deep into the barblus, right, you can see something called as hooklets. Okay, so rachis, extension of rachis, this is the rachis. Extension of rachis is called as barbs, and extension of barbs is called as barblus, and extension of barblus is called as hooklets. So this is the basic structure of a feather. Okay, you take any feather, this would be the basic structure of the feather. And coming to the classification of the feathers, in general, not in specific to African lovebirds, but in general, birds have these 5 types of feathers. Okay, so one is the down feathers. This down feathers is mainly on the young birds. You people might have seen when the bird hatches. You can see some kind of feathers which does not have any structures. It will either be gray or white based on some kind of mutation. The colors may vary from each other. Okay, so this is basically covered in the young birds, and it is randomly scattered. It does not have any structure. If you see, compared to this feather, right, this down feathers does not have any structure. It is just randomly scattered, and it does not have any shape. And one thing to keep in mind is there is no influence on color of this bird. So these feathers will never give color to your birds. You can see it is just a gray color or whitish gray color that will be present in the birds. Okay. So coming to the second and the third type, that is phyloplume and bristle. So these two feathers are hair-like structures. They have barbs, what I showed here, right? They have some kind of barbs at the bottom and the top. These two feathers we don't see in Agapornis, maybe in peacocks or something like that, in maybe other birds also. I'm not very sure where exactly that, but however, let us know that it is not there in the African lovebirds. And next is semi-plume. This one is more structured when you compare to the down feathers. It has a clear rachis, at least a visible rachis when you compare to the down feathers. Okay, and with some barbs, it provides insulation to the down feathers along with the down feathers. So the main function of this feather and this feather is to provide insulation to the birds. both these feathers will not determine the color of the bird. So now coming to the tail feathers and the flight feathers, these are basically called as counter feathers. Okay, so this has the clear ratchets, barbs, barbelows, and hooklets, what I explained here, right? So these two feathers, the tail feathers as well as the flight feathers, has clear indication of ratchets, barbs, barbelows, and the hooklets. So these are the two birds which actually determines the appearance of the bird and the color of the bird. So this is about the basic types of the feathers and the anatomy of the feathers. So now, since the semi-plumage, this one and the down feathers is actually not giving color to the African lovebirds, we will not talk about these two birds, these two feathers. We will only concentrate on the contour feathers, which is the tail feather and the flight feather in the coming slides.
Okay.
So the counter feathers basically is classified into three types. So one is the feather of the structural type, second is a feather of the general type, and third is a feather of the ornamental type. So before this, now let us remember this point. All of you, I think you should keep this in your mind. The pigment in African lovebirds are from citizens and eumelanins. These two words are very important when we talk about the feather structure, and further when we go to understand any mutations, right? So these two words are very important. That is the citizen and eumelanin. So these are the two things which actually gives color to the African lovebirds. Okay, how it will give, I'll explain in the subsequent slides. So as I told you, it is the counter feathers are classified into three types. So first one is the structural type. This is also very important. Keep this in your mind. Structural type. Okay. So this structural type counter feathers is basically blue or green. It will either be available in blue or green. Okay. So this is the cross section of the feather. So the cross section of the feather, you can see the outer ring is yellow. Okay, this is called as a cortex, and it is yellow. And this yellow color is because of the citizen pigmentation. Okay, as I told you here, citizen. This is the pigment. Okay, so this is the citizen pigment that is available in the outer circle of the cross section of the feather. So when you come to the middle ring, so this ring, in photo it is blue, but just for explanation, I'll tell that it is a spongy, colorless, tube-like structure. So there will be some structure in this area which will be little spongy, okay, and it is colorless. I'll tell you why it will be blue in the later stages, but for now you can assume that this is a colorless structure which is there in the blue area. And the next one is the inner ring. So this inner ring basically consists of eumelanins. I think most of you have heard of this term, eumelanin reduction, or maybe eumelanin as a word as such, only you might have heard about it. So this inner circle basically consists of the eumelanins. Okay. So the example, one of the example for feather of structural type is green fisher. We have green color feathers, right? We can consider this as one of the examples for feather of the structural type. Okay. So next is coming to feather of general type. Basically, this does not have the spongy layer. This will not, the general type will not have the spongy layer. Okay. Since the spongy layer is not there, there will not be any color available for this. And this basically is not available in African lovebird. Okay. So we can maybe ignore this. This can be found in finches and cocktails. And finally, it is the feather of the ornamental type. So the feather of the ornamental type will not have the spongy layer. Okay, the spongy layer, whatever is there outside, will not be there. It will not contain eumelanins. Okay, whatever you see eumelanins here, right? It will not be there in this one. Okay, and these feathers are basically it varies from orange to red feathers. So in the birds, what in African lovebirds, wherever you see blue or green feathers, it is basically the feather of the structural type, and wherever you see red or orangish kind of feathers, it is basically the feather of the ornamental type. Okay,
Okay.
so When I explained here, I told you this blue area will be white, but why it is shown as blue, I will explain you in the next slide. Okay. So why now let us understand why this counter feathers is actually blue or green or any other color that we see on the birds. So this is the cross section of the feather. So as I told you, this is a tube-like structure, spongy tube-like structure, which will be colorless. Okay, but what happens when you put light on these feathers? So when the light or the sunlight falls or any other light falls on the feather, some portion of the light is absorbed by the eumelanin. Since eumelanin is black in color, the light is absorbed by the eumelanins. Okay, when the light is absorbed and during the absorption, there is some reflection also that happens. Since this spongy, colorless tube-like structure, it is a structure, right? You have different like a honeycomb structure. So what happens here is some of the light gets reflected. So when this light passes through this structure, right, it gets reflected, and this reflection will cause a blue color light to be produced in the spongy layer. Okay. So due to the tube-like structure, light is reflected in the spongy layer and blue light is created. So ideally it will be white, but when the light falls on the feather, this portion becomes blue color because of the reflection or refraction or interference or transmission, whatever you can say it, if you talk on physics terms. Okay, and this one is the black eumelanin, which absorbs most of the light. So now what happens, we have yellow color citizen in the outer layer and we have a blue color light in the spongy layer. Okay, so when you combine yellow light and blue light, we basically get a green color light, or we will be able to see only green color light. So this is how a feather of a green color feather looks like when the light is falling onto it. Okay, so as I told you, the combination of the blue light and the yellow citizen. Okay, this is the yellow citizen and this is the blue light that is being created, and the combination of these two will result in a green light reflecting from the feather. That is why we see the green color on the feathers of the African lovebirds. And the structure of the feather is decides the color of the feather. So the name feather of the structural type. So now as I told you, there is some tube like structure inside this. Okay, so if this tube like structure changes, the color that will be reflected also changes. For example, in this case it is blue, but if I take an example of a violet factor bird, what happens is the structure inside this changes, because of which a violet light is reflected. Okay, so that time you will be able to see a violet color on the bird. So since the structure inside the spongy layer decides the color of the feathers, it is called as the structural type. Hope this is understood by everyone. If you have any doubts, maybe you can clarify now or towards the end also. Okay, maybe I'll go to the next slide and we can clarify towards the end. Okay, so I'll give some of the examples where we can understand how is the green, blue, violet, and turquoise feathers. So basically, in the green feather, as I told you in the previously, we have yellow citizens and blue light. So combination of these two will give you a green color feather. So in D green, what happens is the structure inside changes. Okay, so because the structure changes, the blue light becomes more darker. So the combination of dark blue and yellow becomes a D green. And in DD green, again, the structure inside the spongy layer changes. And because of this heavily dark blue light that is reflected from the feather and the combination of the citizen, it becomes DD green. Okay. And similarly, in blue feathers, so what happens in blue feathers is the blue light is always there as when you see the green one, right? The blue light is always there. But in this case, the citizen is reduction has happened here. So when the citizen reduction has happened, there is no combination of blue and yellow. There is combination of only blue and white. So when there is combination of blue and white, you can see a blue color feather on the bird. Similarly, when you see a turquoise bird, right, what happens is the citizen, whatever is there in the outer circle, there is slight reduction. Okay. So when there is slight reduction, the combination of a lightly reduced yellow and blue color will give you a turquoise kind of color on the bird. And a violet bird also, you can see that because of the structural changes in the inner spongy layer, there is a violet light that has been created. And because of no citizen present, yellow citizen present in the outer layer, the combination of these two will result in a violet bird. Okay. You may ask what happens, whatever I'm showing here is a blue violet, but what happens in a green violet? So green violet, basically what happens is the yellow citizens will be present here. So you can always differentiate a normal green bird and a green violet bird. Okay, so when you open the feathers, you can definitely see some kind of violet markings on the bird. So that would be the difference between a blue violet and a green violet.
Okay.
Okay, so this is about the feather structures. Now, one more important thing is the classification of mutation. Classification of mutation is basically done on there are some parameters or some pigmentation reduction or increase that is happening, which is causing the mutation in the birds. Okay, so we classify basically the mutations into eumelanin mutation, citizen mutation, feather structure mutation, and pigment distribution mutation. Okay, so eumelanin mutation, basically there is either decrease or increase in the eumelanins, that is this one inside. Either this will be very dark or this will be very light because of the increase and decrease of the eumelanins. Okay, and the second is citizen mutation. This mutation is caused when there is increase or decrease in the citizens in the feathers. Okay, so that is this yellow color, whatever you see here. If there is increase or decrease in this layer or the color of the citizens in this layer, then such mutations are categorized under citizen mutations. Okay, so in eumelanin mutations, we have all these NSL, INO, pastel, dark eye clear, bronze fallow, pallid, marble, pale fallow, dun fallow, peed, U-wings, misty. Okay, and in citizen mutations, we have blue color, we have aqua, we have turquoise, we have orange mask pale, and feather structure mutation. So this feather structure mutation is classified because, as I told you, in the spongy layer, right? If the structure changes, the color that is reflected from the spongy layer also changes. So when there is changes in the mutation with respect to the feather structure, right? So then it is classified under feather structure mutation. So dark factor, violet factor, stylic, crusted, pale-headed, all this comes under feather structure mutations. And finally, it is a pigment distribution mutation. We currently have only opaline under this category. So what this does is it moves the pigmentation only to some specific area. For example, if you take an opaline, for head there is only one kind of color that is being pushed, and for the body there is only one kind of color pushed. So it is basically the distribution. Okay. So now, hopefully everyone has understood. Just to make it clear, I'll once again just summarize. So any reduction, increase or decrease in the citizens, which results into a mutation, is classified under eumelanin mutation. It is classified under citizen mutation. Any increase or decrease in the color in the eumelanins is classified under eumelanin mutations. And any changes in the structure of the spongy layer, which results in change in the color that has been reflected, will result in structural mutations. And finally, it is the pigment distribution mutation where we have opalines. Okay. So now many people have questions that which mutations we have to combine and which mutations we should not combine. Okay. So this will give a clear picture on which we can do. It is not like we should not do. We can do it, but doing so will result in some kind of birds which will not be we will not be able to describe such kind of birds. Okay. So I'll just explain which combinations are good and which combinations are bad in this case. So eumelanin mutation, you take any of these mutation. So it is always better not to combine eumelanin mutation, as these mutations in is unrecognizable. For example, you combine NSL INO with a pale fallow. How will you know what kind of bird is it? Okay, because even NSLNO is reducing the eumelanins in the bird, and pale fallow is also reducing the eumelanins in the bird. So this combination is actually bad, and you cannot differentiate what kind of bird is this. Okay. So that is the reason they tell that eumelanin mutations should not be combined. And coming to citizen mutation, we have blue, aqua, turquoise, orange mask, and pale head. This also should be avoided. Okay. So since blue is the allele, aqua is the allele of blue, and turquoise is the allele of blue, combination of any of these three birds will result in blue aqua, blue turquoise, or aqua turquoise. You will not get a firm bird. It is always an intermediate bird. And blue orange mask, though I don't think so you can combine, and blue and pale head also there is no way that can be combined, because these are in the different species sectors. Okay. And finally, it is the feather structure mutation. We have dark factor, violet, stally, crested, and pale headed. So this can be combined. Okay, you can combine a dark factor with violet. Okay, so you can also combine two dark factors or two violet factors with each other, and you will really get a good bird. For example, you take a DD blue bird and have one violet factor in it. This bird will really look good. Okay, so this combination is possible, and these can also be combined with eumelanin and citizen mutation. So whatever mutation is available under feather structure mutation, you can always combine with the eumelanin mutation, and you can combine these things with the citizen mutation also. For example, you have blue with a violet factor, you have blue with a dark factor. Okay, you have blue with NSLino, that is albino. Okay, you have blue with pale fallow, that is blue pale fallow. So you can combine these things. You have a dark factor, you have a blue, you have dark factor, and then you have maybe you can take Ewing. Okay, so these three combinations is possible. Okay, these two combinations are possible, these two combinations are possible, but never mix these mutations together, or never mix these mutations together. Okay. And finally, it is the the last mutation which is available. So oplines also, you can pair it with either eumelanin mutation or citizen mutation or feather mutation, or all of the three together, or all of the two together. So opline works well with all the other mutations that is available. Okay, so this is about the combination. So hope everyone has got a fair idea on what combinations is good and what combinations should not be done. Okay.
Yeah.
So this, I have just made a matrix on what combinations can be done and what combinations should not be done. So maybe once I share this document to you people, you can keep this as a reference. So as I told you, eumelanin cannot be combined. Okay. And citizen and citizen mutations cannot be combined. So which other combinations is there is always welcome. You can always combine these mutations and get really good birds.
Sure.
Okay, now finally coming to the mutation, NSL mutation as such. So now I think you will be able to understand this mutation in a much better way. Why the head looks red and why the body looks yellow. So as I explain, you will get to know why these things happen in the birds. Okay. So NSL is one of the most common mutation. I think most of you in your AV areas have INO mutations. So this is one of the most commonly found in African lovebirds. It is an autosomal recessive mutation. Okay. In my first session, I had explained you what is an autosomal recessive mutation, which means to say that both the genes, this mutation should be available in both the genes to produce the visual characteristics of NSL INO. Okay. So since it is a recessive mutation, and this is classified under eumelanin mutation. Today I explained about the classification, right? So this is a eumelanin mutation, and it is actually a reduction that is happening in the eumelanin. Eumelanin mutation either then can be increase in the eumelanins or decrease in the eumelanins, but in NSLNO it is actually the reduction of the eumelanins that is happening. Okay, and in this mutation, eumelanins not only disappears from the feathers, but also the feet, nail, and the eyes. You can see the feet, you can see the nails, and you can see the eyes. Because of the eumelanins disappearing, all these colors have been, the colors on all these body parts have been affected. And due to eumelanin reduction, the feet and the nail turns horn color. Like we have cow's horn color, right? When we cut it, we can see a creamish pale color. So you will see such kind of colors on the toes and the nails. And since the black color, so basically eumelanin reduction is black color reduction in the bird. So now the black color is also reduced from the eyes, so you can see only the red color in the eyes. So this is a brief introduction to the INO mutation. So now we'll understand the feather structure of the INO mutation. So as I told you, this falls under eumelanin reduction. Okay, so there are no eumelanins. Okay, since there are no eumelanins, once the light falls on the feather, okay, there is no absorption of the light is happening. You must be knowing that black color absorbs the most light. Okay, so since there is no eumelanin, there is no dark color that is present here. Since there is no dark color, there is no light absorption that is happening. Since there is no light absorption that is happening, there is no blue color or blue color light that has been created in the spongy layer. Okay, so now since the blue color is not available, we have only the yellow citizens that is available on the outer circle. So this results in all the green feathers turning into yellow feathers. So if you see the NSLino bird, basically you have in green fisher you have some kind of dark patch here, and you have green color on the body as well. So here what has happened, since the blue light is not reflected, you have only the yellow light that has been there in the feathers. So wherever there is green color, it all turns into a little bit yellowish or even white. I'll tell you why white in the next diagram. Okay. So basically in white, what happens is there is a reduction of eumelanins, okay, which will not absorb the light. So this will become white. And in outer layer, there is no citizens also. Okay. So this will also be white. So this is the reason why an albino looks like a white color bird. Okay, so albino is basically a combination of NSLNO mutation and blue mutation. So as I explained you previously, in NSLNO mutation, what happens is there is no blue color light that has been created, and if we combine it with blue, so when we combine it blue, there is no citizens also because blue comes under citizen mutation. So there is no citizen also. So this results in a white color feathers on the albino.
Okay.
Okay. So now coming to the breeding of NSL Lino, as you may know, breeding of NSL Linos is not very easy. Most of you would have tried. I have also tried and kept it for 7 to 8 years, and it was very difficult to breed these birds. Not like the fishers where you get 5 chicks or 6 chicks. Okay, and chicks usually die when shortly after the birth. So within 1 or 2 days, they just die. This happens because they are very weak, and even the younger birds, if it is slightly bigger than the newly hatched birds, they will just stamp each other, and this will result to the death of the bird. It is very important to pay attention to the good quality of the birds. Whenever there is a eumelanin reduction kind of mutations, right, it is always better to pick up good quality parents. Okay, for a case of for in case NSLinos and pale fallows, etc. So here, because here the eumelanins are reducing. Humulin reduction is actually not a good what do you say characteristics. Okay, even in human beings, when the reduces, you can see that some people will have white patches on their body, on the skin. Okay, so that is because of the humulin reduction, and direct sun rays falling onto such kind of skin, right, which will lead to the deterioration of the skin. So it is actually not good when the humulin reduces, but however, since we are keeping these birds in a controlled environment, it should be okay. Okay. So never pair the birds with closely related birds. This I think most of you know. And also, I would not consider pairing NSLinos and NSLinos together. Okay, with my experience of 7 to 8 years keeping INO mutations, it is very hard to get more than one or two or three chicks when you combine such kind of when you do INO into INO pairing. Yeah, there will be some cases where you will get 4 or 5 birds, but pairing these two things, no, it is not only on the number of the birds. Once the chick grows, right, they will not be as good as the parents. Okay, they'll basically be weak. So better not to pair Enos with Enos, and always pair the below three combinations if you want to produce a really good birds. For example, first you pair it with a wild type to get a very good wild type split and then you can always combine the wild type split to get a bird, or you can always use one bird and a split bird to create a good or to produce a good bird. Okay. So now, till here we talked about only the NSLino mutation, okay, which is a combination with the wild type. That means that the green type. Okay, so this is the green series NSLino bird. But what happens when we combine NSLino with the blue mutation? Okay, so when ENO is bred into blue mutations, all the citizens disappears. Okay. So in previous, you can see there is no blue color, humulin reduction, and only the yellow part is there. Okay. But when you combine it with blue mutation, blue is basically a citizen reduction mutation. Okay. So here, what happens? The citizen is completely reduced. The yellow color is completely gone. So now what happens? All the feather becomes a white color. So that is why an albino looks completely a white color. Okay, because of lack of citizens and eumillinins. There is no citizens, there is no eumillinins. This will result in a white bird. Citizen reduction makes the red beak into horn color. You would have seen that the albinos will not have a red color, because as I told you previously, citizen is the pigmentation which gives red or orange color to the bird. And some combinations for albinos are NSL green into blue. You can create a green split blue split bird from this. And then you can combine some of the splits, for example, NS split blue into NSL split blue birds to get a albino bird. But always keep in mind that visually there is no difference between NSL blue mutations in different species. For example, you take fisher, you take Sonatas linearly or Niger genus. All the bird will look like a same bird. So when such kind of ambiguity is there, when you're not able to decide what kind of bird is it, such kind of birds are always not entertained in the shows. Okay. So keep in mind that I think albinos is not accepted in most of the shows, and even some kind of ENO or deck ENO, some kind of combinations like this are always not accepted in the shows. Okay, because they will not be able to judge what bird is this, whether it is a fisher, a personata, Liliani. So they'll not be able to judge this. So that is the reason it is always not entertained in the bird shows. But yes, definitely Enos have a very good commercial values. So nothing wrong in keeping Enos. So now when we combine Enos with turquoise or parblue, okay. So here what happens is when it is bred into parblue, there is a partial citizen reduction. So what happened in albino is there was complete citizen reduction. Okay, the albino blue is always called as a complete citizen reduction. There is no citizen. So That is why it has become white. But when you combine it with a par blue or a turquoise bird, a turquoise mutation is basically a partial reduction. There is no complete reduction of the citizens. So when there is partial reduction of the citizens, the yellow becomes a pale yellow, and this is what results in a pale color of the bird. And since we have combined the INO mutation with the turquoise, we get a like characteristics in rest of the parts, that is eyes and the body and the legs. Okay. So here also some of the combinations which you can use is NSL green into par blue. You will get a green split split par blue birds. Okay, so these birds you can combine again to get a par blue bird or a turquoise bird. So this is about combining with Enos with turquoise. Okay. Okay, so this is till here I explained about the enos, and once you put this enos into green, blue, or turquoise, how the feather structure changes and what are the characteristics of the birds. Now I'll explain what is pastel and why it is called as a allele of Anasilino. Okay, or maybe why it is called I'll explain later. But pastel is called as the allele of NSL Eno. Okay, I just showed here, right? So NSL Eno is there, pastel is there, dark eye clear is there, that is deck is there, and bronze fallow is there. So all these mutations are called as the alleles of NSL Eno. Why, I'll explain you later. Here you can see I've just put a star bronze fallow. This still there is some confusion in understanding this mutation, but however, let us talk only about the pastel and dark clear. So in pastel, what happens is this is commonly known as yellow fisher in African lovebirds. Pastel is an allele of NSL Eno, as I mentioned. Okay, this is also an autosomal recessive mutation. And since it is an autosomal recessive mutation, we want these mutations to be present on both the genes to produce the characteristics. This is again classified under eumelanin mutation. Okay. In this mutation, eumelanin reduction is about 50%, and it affects the feathers and the feathers, but also the feet and the nails. Okay. So here we can see that the eumelanin reduction, where the green. Okay, so this is the green feather, right? So there is 50% eumelanin reduction. Okay, so in enos, what you see is 99% eumelanin reduction is there. So there is nothing at all here. But in pastel, what happens is there is 50% reduction of eumelanins. So that is why it is not a very vibrant green that you can see as in the normal fisher. Okay, and you can see a slight reduction, or there is a light shade when you compare to the normal wild type. Okay. However, the eyes in this case will remain the same. So these two are the representations of a pastel green bird and a pastel blue bird. So it is kind of a dull bird, which when compared to the wild type.
Sushil, do you have any queries? Hello, Sushil? Okay, yeah, Mahen, you can go ahead.
Okay. Okay, so this is about pastel. Nothing much to explain here. There's just that you have to remember that there is a 50% reduction in eumelanins, which has caused this bird to be a little lighter. Okay. So next we have dark eye clear, which is also called as headache, and this is also an allele of NSL. So dark eye clear is basically called as bull in our local language. I think most of them use this name in African lovebirds. So deck is an allele, again, as I told you, it is an allele of NSL. This is also an autosomal recessive mutation, and it is classified under eumelanin mutation. Okay, in this mutation, the eumelanin reduction is 95%, and okay, and it affects the feathers, feet, nails, but however, the eyes remain the same. So now you can see the difference. You can see here there is 50% reduction in eumelanins. So still you can make some differentiation between the oranges or the greens and all those things. But when you come to deck, right, there is 95% eumelanin reduction. Okay, so this almost looks like a bird. If you see both the blue series as well as the green series, it almost looks like a bird. But the only difference between an bird and a deck bird is you will have a dark eye. Okay, will have a red eye, and you will have a rump which is blue in both the green series bird and the blue series bird when compared to the bird. Okay, so this is the difference between and the deck. Okay, so just to summarize, NSL mutation, there is almost 99% reduction of eumelanins. Pastel, there is deck, there is 95% reduction in eumelanins, and pastel, there is around 50% reduction in eumelanins. And all these three are called as alleles of each other. And finally, we will understand why it is called as alleles of each other and what happens when you combine these birds. Okay. So I think this discussion is going on, and there is a lot of questions about this in many hobbyists. Okay, what happens when you combine pastel with a deck with and all those things. So we get an intermediate bird. Why we get an intermediate bird, I would like to explain. Okay, in the first seminar, I explained there is a cell, there is a nucleus, we have chromosomes, and then we have the DNA, which actually carries what kind of bird has to be produced from this cell. Okay, so this is a gene. Let us consider that a small change in this gene has resulted in NSL-INO. Okay, again, a small deviation from NSL-INO has caused a production of a pastel bird, and small changes in the same gene has resulted in a deck bird. Okay, so whenever there is a change in the same gene which is resulting to different mutation, right? For example, if you consider this ABCD, next time it will be aligned as BCAD, and next time it will be some other combinations. So at the same location or at the same gene, when there is a different modification that is happening at the same location, then they are called as alleles of each other. Okay, so now in NSL, basically there is a modification in locus A. They call it as location A. Okay, in DNA there are a lot of locations that has been given to each of the genes. Okay, so now let us consider this is location A. Okay, whatever you are seeing here, this is location A in which there is a gene, and any change to this gene is causing NSL mutation. And the same gene, if there is some other kind of changes, it is causing a deck, and some other kind of changes, it is causing pastel. Okay, so in the same gene, when there are variations and it is causing different mutations, it is called as alleles of each other or alleles of each other. Yeah. Okay, so now NSL, deck and pastel are called as alleles of each other because it is a modification of the same gene in three different ways. Okay, and combination of such kind of mutations will always result in an intermediate bird. You will not get a split bird. You when you combine with a deck bird, you will not get split deck or deck split something like that. It will always be a combination of into deck will be deck combination of into pastel will be pastel and combinations of deck into pastel will be pastel deck. Okay, so this is one of the photo which I picked up from one of the hobbyists. Okay, so you can see the difference. This is a clear deck bird. Okay, you can see there is some blues, and also in some places you can see the shades of blue. Okay, so here when you combine with the deck, right? So what happens is again you will get an intermediate bird which is in between the deck and Okay, so in deck there is blue, and in you can see that it is a completely white bird. So when you compare these two, you will get an intermediate bird between these two where there will be a reduced white color. This is actually a decino bird. Okay, So you can see that the blue colors have reduced. The rump is also not blue in this case. Okay, so it almost looks like an INO bird, but the eyes is red. Okay. So the final point that you have to remember is combinations of alleles do not produce split birds, but it will remain in an intermediate bird. There is one more combination similar to this. If you have noticed, so we have blue, we have aqua and turquoise. So aqua and turquoise is basically alleles of blue mutation. Okay, so whenever you combine blue and aqua, you should always tell that it is blue aqua bird. You cannot tell it is blue split aqua. Or when you combine a blue bird with a turquoise bird, it is blue turquoise. It is not blue split turquoise. Okay. So whenever the alleles are there, you have to always use the both the mutations names in a single word. I think that's it for today. So any doubts or clarifications which you want to do, I would like to answer those questions.
Like more technical, man. To understand the slide itself, it will take me 2 days.
Yeah, I think, but yeah, I took almost maybe months to understand all these concepts. But once you understand these concepts, right, you will really enjoy the hobby, like why these colors are coming, why it is reducing, why it is increasing.
Mm.
So definitely, yeah.
Okay. Yeah, any doubt, guys? Like you can. It's not about like this topic. If you want to
No,
clarify any
you can ask
questions.
any doubt about the mutations or whatever it is. You can try to clarify. Not sure if it was very technical and it was difficult to understand. It's okay. Maybe we can share this to
Yeah,
each one
slide
of
and then
you. Yeah, and try to understand. See, actually studying this from the entire article or the book is very difficult. Okay, you will take months together to understand these concepts. Just try to put it in a simpler way so that it can be understood just by seeing the slides.
Okay.
Yeah, I think someone has raised
Sushil.
their hand. Sushil,
Yeah,
yeah.
Sushil, I asked, but
Okay.
yeah, find my no problem. Yeah. Guys, like if you don't have any questions, you can wrap up, or you have any mutation or related questions or breeding related questions to
Yeah.
breeding combinations, you can ask for INO and other things. Yeah.
Hello.
Yeah, Mohammed. Yeah, sure.
Sir, I just a doubt.
Yeah,
How to
sure.
identify albino opaline visually? How to identify albino opaline and normal albino?
I think as far as I know, I don't think so. You can differentiate albino with any other mutation. It is very difficult. So
We'll
that is
send that
that.
to
Yeah.
parent details if you know that only
Yeah,
we can
only
let them know.
only if there is only if you know the parents, then you can say whether it is an opline or not an opline. So as I told you, no, so albinos are never considered as a good bird for any of the shows. They don't accept these birds because every albino bird looks the same. Whether it is combination of different mutations or species or whatever it is, it just looks same. So it is very difficult to differentiate albino mutations in terms of species as well as different combinations. So hope this answers your question.
Okay. Thank you, sir.
Right.
Mahenji,
So when
may I
people
ask
are
one question?
yeah, yes,
I'm
Mohammed.
Amal. My doubt
Yeah.
is today you discussed and we learned a lot about these mutations. Okay, my simple
Hmm.
doubt is,
Hmm.
especially I'm asking about in eye species like our fishes and masks.
Hmm.
The pairing of means lutino fishes, albino fishes, or parblue
Hmm.
fishes like that. Pairing What is
Hmm.
your suggestion? Because I have seen many of our breeders, they are breeding this into
Hmm.
So what is
So
your suggestion? So is there any guideline that we should do or we should not do like that?
okay. So what I used to do, in last 7 years, what I've done for Enos is, of course, I'm not keeping Enos anymore, but I have kept this for at least 7 years. Okay. So what I used to do is whenever I used to pair, right, I used to pair Eno into Eno. Okay. Then check how is the outcome. How is the outcome of the birds? Whether there are huge mortalities or the chicks are dying in the shell. So I used to just record all these things and understand. For example, if I have kept 5 different pairs of Enos into Enos, there will be at least one pair or sometimes no pair or one or two pairs which gives good chicks, maybe 2 or 3 chicks, and the size is also good. They're very healthy and all those things. But definitely, there will be 70 to 80% of the birds that you have paired will not work out well. For example, if you keep 10 pairs of Enos into Enos, you will hardly get 1 or 2 pairs which is good. They give you good babies. The babies are healthy, and the rest is not good. So what I do is in such cases, I also parallelly work with splits so that I can combine those splits with these birds. So that is how I started getting more productivity in terms of Enos. So whenever you first always try into if you have the liberty and time to do it, right, always try with into check if the chicks are good or the quality is good. If it is not good, then definitely you can pair it with a split bird. So that is how I worked out with getting good birds from Enos into and
Okay,
people.
okay. My major doubt is you described very well, but the thing is, my another doubt is
Mm,
if we pair into as you
mm,
said, in 70-80 percentage, we may get a good result. That means the outcome, we will get chicks, enough chicks. That's for a breeder, that is enough and more. Then what will be the future of those chicks? And if somebody buy the same chicks,
mm,
that chicks, okay.
mm, mm,
If they do the same, means if they buy the chicks and they will also do the same, means
mm,
into what will
mm.
be the result for another 3, 4, or 5 steps after? What will be the result? Because I have seen many Enos coming from the same way. When they are around 5, 6, or 7 month age, they will
Mm.
become weak. They may show
Yes,
so many illness
yes,
without showing any what to tell symptom, or even if there is no infection is there, even then the birds know they will become ill. They will die very soon.
yes.
And
Yeah,
the
even
fertility part also,
yes,
pairing part also, they will be the worst birds.
yes, yes, yes. Even I have experienced the same. I think I've bred 3 generations of Enos. So I'll definitely tell you that the first generation of Enos were not same as the third generation of Enos because it was Eno into Eno, Eno into Eno, and Eno into Eno. So the quality of the birds or the size of the birds will definitely keep coming down. The only suggestion is it may be any mutation, not only Enos, right? Always once you get a bird, pair it with a wild type, create splits, and again then try to pair it up with a mutation so that you get good birds. So every generation you pass on, it is very much required to pair it with a wild bird so that the wild type or the wild genes is passed on from one generation to another generation. So that is the
Because
situation.
why I asked this question, no.
Hmm.
One thing I'm very happy to tell you in Kerala. I'm not telling about anything bad about other state. One
Hmm.
thing I have seen in Kerala, if you ask 100 people,
Hmm.
99 people are pairing, especially the fish in fisheries and all hiring birds.
Hmm.
Everybody is pairing
Hmm.
one with
Hmm.
an split. That
Hmm.
I have seen in Kerala.
Yes,
Whether
yes,
they are educated, whether they are rich, poor, that doesn't matter.
correct.
Everybody is pairing because of the, I think, learning and all.
Yes.
People are pairing only like that. Even in the market also, chicks are also available like that.
Hmm.
Enos, then
Hmm.
Enos
Hmm.
splits like that.
Hmm. Hmm.
And now the thing is, I think people are running behind this albinos. The recent scenario last
Correct.
2, 3 months, The out of Kerala people, everybody is asking only
albinos.
albino. Nobody needs That's why I noticed this phenomenon. Okay, everybody is telling that they are very crazy. They are very. They try to learn genetics, everything, standards, blah blah. But at the end of the day, people will pair like that only. That's
Yes,
it.
yes.
Just asking.
Yeah. That is there. I think that is one thing we might have to change among serious hobbies. Yes, commercial part is one way, but if we really want to lead in this hobby, right, I think some changes has to be done. At least some people have to take up this responsibility of producing good birds. The only reason why albino not many people question and take it is because there is no differentiation. For example, you take an opline, and if it is a hybrid of a personata, you can always see a mark, and people will immediately reject it. This opline is of not good quality. Okay, but in albinos and lutinos, most of the cases, it is a crossbreed, hybrid, whatever breed it is, it looks the same, and it sells also pretty much quickly. So yeah, that is the situation. Yes, there is commercial aspect to it, but if you want to understand about this mutation, then if you want to produce good birds with good size, then always better to do it with splits.
Yeah, mine one more.
Okay, Mahin, thank you.
Yeah.
Comment, somebody posted Abhijit,
Yeah.
Olive. Can I click the comment? Yeah.
Sorry, I'm not able to see the comment.
Messages.
Okay.
Next to it.
Got it.
I think in your second slide you discussed, right, about this olive, the dark factor will have the more blue in the
Yes.
illuminate. Yeah. So if we mix the mauve turquoise, so what will be the output like? We
Okay,
get
so
the more dark factor of violet or blue.
okay, let's go to the slide and try to
Third slide,
understand.
right?
Yeah. Okay.
Third
So
or no, before the slide, I think.
26th slide, if I'm
25.
25, this one, right?
Yes.
Okay.
I think the question is related to this. Yeah.
Okay, so he's asking, explain what is the pairing good
Olive
or
is
bad? Okay.
olive in the sense a double dark factor green.
Yeah. So he is telling he has a double factor. Okay. Mevu also means that I think it is
Double dark factor.
double factor. Okay. Or sorry, olive is maybe single factor and Mevu is double factor. Okay. So there are two factors. This is fine. We have peach and turquoise. That is green and blue. Yeah, this is absolutely fine, I guess. Only thing is you will get a green bird.
Green bird, right? Yeah.
You will get a green bird with some birds with one dark factor and some birds with two dark factors. Yeah, this is okay. No problem, I feel.
So any other doubt, guys? Like, one you can ask. Hey, Rahul. Yeah, I think Rahul joined. Okay.
Okay. Yeah, I understand the silence because it's difficult to understand the concept.
I'll do mine. Like, no, let me share your PPT. I'll go through them, and then if required, we can conduct one more session also.
Yeah, we can clarify this doubt. See, as I told you, no, these things cannot be understood in a half an hour session or a 1 hour session. It takes time. My only request to all of you is please go through it, understand, so that if you will be better confident of yourself when you are producing birds or when you are selling the birds. That's it.
Okay, fine. Yeah.
If nothing else, then we can. Okay, I think we have one more question. Okay, if double factor blue violet and double factor blue violet both are split INOs. Okay, so there is NSL INO coming, which carries violet factor. Okay, is this possible? Yes, I think. See, whenever there is a blue INO, right? INO in a blue series. Okay. Violet factor will actually not contribute more, so it will still look like a albino bird. Hope that is clarified. Even though the violet factor is there, you will not be able to see this violet factor. So it'll just look like a normal albino bird only. Hope I've understood the question properly and answered. Muthu, can you if
Yeah.
double? Okay, right?
Yeah, double dark factor blue violet. Both are split INO, so it will produce INO. Yeah.
Yeah.
So
and he's asking if it carries the violet factor. Yes, it is possible, but it is an albino bird. You will not have much influence of.
reduced, right?
Yes, you will not have the violet to be seen on the bird.
If any doubt, if you want, you can ask. It's related to this.
Hello.
Yeah, Furkan.
Hi, Mahen. Hi, Muthu.
Hi.
I just wanted to know. I know about them. I know about it, but I just want to just have a take with you guys. So I have two splits of fallows. So
Two?
I have a split fallows, split
Okay.
INO.
Okay.
Okay. So I have a pair of fallows which I bought quite a time back, and I knew that I had to not breed them as in to get good results, but I wanted to take a chance.
Okay.
And luckily, I was lucky to get enough, and I bred them. So
Okay.
now I am left with two splits.
Okay.
So if I'm pairing both the splits,
Yeah.
what would be the result?
So
Like
if you're
how many how good would be the results?
it'll be I think there will be some visual pale fallows. Okay, there will be some
Split
some splits of pale fallows, and there will be normal
Possible.
pictures. Yeah, possible. You can just say possible. You
In
cannot
that
answer.
normal and possible. So, is there
Yes.
a high chance that I can get fallow on this, or it will be
Yes,
how is it?
definitely. Yeah, if you're putting two splits, you have, I think, 25% chances of getting fallows.
So
Furkan,
the rest we have
people,
to keep and test breed it.
Yeah,
people will avoid the
yes.
fallow to fallow because of the illuminant is reduced,
Yes,
right? 80%
yes,
of the
yes.
illuminate.
Yes,
So
I know.
again, it's a reason. Again, NSL INO concept. Yeah. So
Yeah, but
that's a reason they'll go for the splits.
I have been keeping birds from quite a time.
Mm-hmm.
It's been about more than a decade, but in this, I have tried my hands nowadays. So
Okay.
there are a lot of people nowadays, a lot of new breeders who come up and they tell, yeah, you get good results. But I wanted to actually know from a person who's worked onto it. Because now, if someone is selling birds, so usually now in the market, everyone is selling a fallow with a split.
Yeah.
So you are ending up buying splits. So if you are trying your luck in terms of fallows and if you have the splits, is it good to breed the splits or it is better to just pair them with the fallows?
Yeah.
It's better to break from the splits, I think. Yeah.
Yes. Both options are fine. Like one, you can either go with split fallows. Okay. But the only question you will have at the end is the birds that are coming. You
Possible.
will which ones are not visual,
So
right? That will be
if I'm
possible.
lucky enough, I'm gonna get the visual birds out of it.
Yeah.
And rest I'm gonna be left with it.
Yeah, it will be possible. So
So
to do a better to increase your percentage of fallows or enos, right? You can always split it with a visual fallow, then a bird with a split fallow. So that time you are clear that whichever is
whatever
non-visual
is
bird
totally
is
splits.
are totally
Ah.
splits. Yeah.
Yes.
So that you can be clear.
Okay. So no, I actually knew this, but I wanted to, have a take on how good it is to pair two birds because the elder pair which is breeding right now, it's with the chicks. So Alhamdulillah till now they are fine. So I don't want to disturb them, and I already have those splits, which are going through the age. So sometimes what happens is I have a lot of Africans with me. So if I have seen in Africans is once they cross their age, their adulthood, and when you don't breed them at time, later they are not easy to breed. They don't pair up easily.
Correct.
So these problems occur. So anyways, breeding split with a normal bird is better. That I thought I'll breed a split with a split.
Yeah, you
But
can
then
do it,
again,
not an issues.
huh,
Only thing is you will have difficulty if you want to sell the bird. You will have
identifying. Yes.
difficulty to sell it. But if you're keeping for your work for the future, then I think it is absolutely fine.
That is there.
Yeah.
So just wanted to check if someone has bred it and how good have the results come. Because usually, if you're buying from a person who's selling you, that's not an honest answer you get 50%
Correct.
of the time.
Correct.
They give the answers which is suitable to their time. Like if you ask them, is it good? They'll be, yeah, 50-50 and 75-75. So I didn't want these answers. I wanted
Okay.
someone who's bred in the group, or it's
Yeah.
an open answer to all the group members if someone has
Yes.
bred it.
Yeah.
So that you get to know what are the possibilities, and also that once you start breeding, then the overcrowding is not happening here. Like you don't keep the possible splits and the splits also, because it keeps on overcrowding.
Correct. Correct.
So anyone in the group who has bred 2 splits and got some
Yeah,
results?
I can tell you from my experience, not the pale fallow, but the INOs. I have bred with green split INO, green split INO. So
Okay.
yes, that you have very less chances of getting like 25% chance of getting like if there is 4 eggs, there is only one bird which you can expect that it is a visual
So
bird.
this case which you have told now,
Hmm.
I wanted to share an experience which I have in my breeding setup.
Hmm.
So quite a time back, I had bought a pair of masks,
Hmm.
which was a known breeder from Bangalore. He had put a black mask, a regular green bird, with a pied fisher.
Hmm.
Mask and fisher he had paired, and
Okay.
in out of that, some birds used to come mask, and some birds used to come fishers, green
Hmm.
fishers.
Hmm.
So I had kept those birds,
Hmm.
and I had a pair of normal blue masks also, which were foster pairs.
Hmm.
So one of those pairs, from the blue mask, I paired one male. So this olive, it was at the start it was an olive mask. Then
Hmm.
later, eventually, when it got to its adulthood, it changed into white fisher.
Hmm.
So now, out of those birds and out of those pairings, I had those 2 pairings which I had given few birds from the same pairing to my friends also. Now,
Hmm.
most of the pairs in them, I'm
Hmm.
getting enos, as in lutinos and albinos. How
Hmm.
is this?
Yeah, either it has to be a split to enos. Okay, that is first thing. If he
Okay,
is
from
getting
one side, I understand.
hmm.
But does it work like if it is only one side also, sometimes you get
No.
splits?
It's not possible. Genetically and scientifically, it is not possible.
Because the blue mask pair which I have, I am very sure I had it from a long time, and I have never got enos from it.
Okay. Okay. Yeah,
And the
that
fisher,
happens. I have seen people who have didn't even know that some split fallows are split to blue. They've got 4 clutches, like 5 clutches without a blue bird. But suddenly, 2 consecutive clutches, they have blue fallows
got a
also
bluebird.
coming out. Yeah.
Okay.
So it's not necessary when we tell 25%, it is just a probability. Okay.
Okay.
So there might be cases where you might get all the green birds with all the splits. Okay. You might.
So in this pair, sometimes what happens is,
Hmm.
at my house, it's about if it is a clutch of 5, there are 3 chicks which are green fishers or a blue mask,
Hmm.
and 2 albinos, or 4 green and blue and 1 lutino.
Hmm.
This has been happening, but since last 2 batches, I haven't got any albinos or lutinos in that.
Yeah, it
At
happens.
my friend's place,
Hmm.
it was one time where all 5 out of 6 were all enos.
Hmm. Hmm. Hmm.
4 were lutinos and 1 was albino.
Yeah.
So it was quite surprising, and the person who bought from me, he was a friend of mine, and he was very happy that a pair of around 3,000 gave him so good chicks.
Yeah, this happens. So yeah, we never know until and unless we don't know about the parents or the grandparents of the birds, right? We never
Okay.
know what genes they are carrying.
So in this case, both the parents might have been possible.
Yes, possible splits, splitting
Okay.
us.
The
Yeah.
blue mask pair also had somewhere a sex link
Yes,
to
yes.
Okay. So now another question. This is was having the question. I forgot now. Again, I remembered.
Hmm.
So I told you the pairing which I had put right.
Hmm.
So now from the INO bird, which I'm getting, from the green pied fisher, I'm getting an albino. The father is blue mask and the female is green pied fisher. I had the same, from the same clutch, I had retained a female, which is again green pied fisher. And I had put it to the same blue mask. The same brothers, both brothers, I had paired it to both sisters. But for some reason, I broke one pair and I just bred one pair. So now I have this green pied fisher and this albino chick. So will it be good if I pair this albino with this green pied fisher?
First thing first, I think you should not pair the siblings. So that is one point.
It is not a sibling.
Okay.
It is from the mother. It's the mother sibling.
Okay.
It's not from the same batch though.
Okay. Even if it is closely related, also I would suggest you not to do. But for your answer, you can pair it, but you will definitely not know what is it split to.
Okay.
It can be a possible split.
Okay.
To any of the combinations that were there in the parents.
Okay.
So only test breeding will tell you whether this has whatever you're looking for.
Because these were a little bit of extra birds which I was having. Currently, people were around openings and those, so I was into those, and now the time is a little bit overlooking too. So I thought, let me experiment with these birds. Like if this birds, the females had carried actually, So if it had carried, so this which I have is strong enough to produce a chick from it, right? The albino.
Yeah, you can.
Okay. That's all. That's all were my doubts.
Right. Thank you.
And I would like to appreciate and thank you for taking so much of time, and it was neatly, presented, and it was very well and neat. And after a long time, I've enjoyed this session of a webinar in this hobby.
Thank you so much.
I would actually appreciate the BKSA team to initiate something like this and to get along all the breeders who are into producing something good and carrying this hobby forward, because nowadays it's just become trade and sale. So I would like to appreciate all the team members.
Thank you.
Thank you for your feedback.
Thank you, Mahen.
Thank you.
That's it.
Guys, any other doubt? I think the mind, like the next topic, can prepare fallow related things.
Yeah, we can explain. Sorry, I don't have much experience in fallows, but the basic concept anyways remains same. So I can conduct it.
The people who are already aware of the mutation or color mutation, so they able to understand the slide very easily, whatever topic we discussed.
Yeah.
But for the new people, like it is really tough, so they have to go through.
Correct.
Yeah, that's good.
Okay.
Any doubt, guys? Like any clarification? Just ask him, like more. If it is not even technical, also we can directly ask color wise or anything you want to clarify.
Any doubts you guys want to clarify, we can do it. If you don't have the answers, also we will get back to you in the later stages. Any questions not related to this topic? Okay, I think Muthu.
Sure.
Conclude. Okay.
Yeah. Okay, Mike. Yeah. Please share the.
I'll share it.
So that I'll share it to the group.
Sure.
Okay, guys. Thank you.
Thanks
for your.
Thank you all.
Thanks a lot,
Thank
man.
you.
Thanks. I'll call you back.
Yeah. Sure. Thank you.
Thanks, man. Bye.